I'll be discussing a bit of the experimental evidences that actually led to the formation of what you're seeing today as molecular concept. This is just some part of the experiment, and some of the experiments will be, the data from other experiments will be shared with by my colleague, Dr. Danny, further by Dr. Ellen. So I'm going to talk on the drug metabolism and dose response aspects, where the journey actually started. So Murali sir was talking yesterday about the MOXPHOS system, that is the oxidative phosphorylation, which is obviously one of the most important thing, respiration, that we have cracked down. And what you see over here on the left hand side is the microsomal system, that is the microsomal xenobiotic metabolism system, metabolism on the left hand side, which shares a lot of similarity with the oxidative phosphorylation. And this is what Sir had talked about yesterday. But then the journey actually started with the MXM system, the microsomal xenobiotic metabolism. So some of the key features that are shared commonly, you can see that the NADH requirement, and then the requirement of the substrate to give it a pull, the thermodynamic pull that is required based on our urban concept, the thermodynamic pull that is actually created because of the presence of the substrate is quite an important stuff. And then further the reaction happens over here, and it's taken up. We'll talk about that in much detail in the coming slides. And a similar thing is observed in the oxidative phosphorylation. Now, yeah, so the thermodynamic pull that we were talking about, this is the experimental proof of the pull that that I have just spoken about. You can see these two different systems. These are peroxidation systems which share a lot of similarity with the microsomal xenobiotic metabolism. Quickly, the reaction is driven. So this proves the role of thermodynamic pull in the presence of substrate. So as long as you don't have it, the dross that we are talking about since yesterday is kind of moderated by the enzymes that we are dealing with. And once there is a substrate, the requirement of that dross is very evidently seen here and is taken up. So yeah. Now let's get into the system a little bit. The microsomal xenobiotic metabolism system, the P450s. For that sake, we are going to talk a lot more on P450s, and there will be some instances where we are going to see the peroxidase system. So here, what are the key players? You can see you need the electron donor. NADH or NADPH is one of the first things that from where the reaction begins. These are some of the partners there. So I'm also talking about the classical mechanism, then we'll refute it, we'll show how that doesn't really work. And then then you need an electron transfer mechanism, which is done by the flavin containing enzyme, which is otherwise known as the CPR, which is a cytochrome P450 reductase. So this reduces the P450. This is the classical understanding. But basically what we are trying to show here is that these are the components of the reactions. Okay. So, and of course, without oxygen, nothing's going to happen, right? But then, what do we talk in urban? We say dross drives the reaction, right? So when you're talking about dross driving the reaction, and the reaction is happening at the sieve, that is for sure, right? So, when you talk about the dross driving the reaction, the dross can be generated here, or it can be generated here, and then can be transferred to the P450 system, right? You don't really need a relay system, a very deterministic relay system for that purpose. So, and yeah, of course, I just missed the cytochrome P5 is again a major modulator of the ROS When you look into the xenobiotic metabolism, you will see P450s are able to to carry forward all of these kinds of reactions. Like you can talk about hydroxylations, you can talk about the nitrogen and sulfur oxidations and epoxidation, dealkylations, all these reactions and many more that it can carry forward. So the thing that I was talking about. Now we are just having a look at the classical understanding and the Merban perspective. So when you talk about the classical system, the reaction begins with the binding of substrate. Now I'll go to the other slide. This is one way of presenting it. Okay. So it actually happens when there is a substrate. See, RH is actually the substrate. So the low spin state of the enzyme requires the substrate to bind here, according to the classical mechanism. And then only after the substrate binds, it can, the whole electron flow can take place, and only then the reduction will happen, and only then there will be an activation of the oxygen. And eventually, all this time, you need the substrate to be bound here, and then the hydroxylation or the reaction that we have seen might actually happen. Now, looking at all this, it really takes a lot of time, and it's not very fruitful way of explaining this kind of a reaction. You get a very low turnover with this kind of an understanding, with this kind of a scheme. But then when you talk about Merban perspective, Merban concept doesn't talk about binding of the substrate here in the first. It talks about the formation of ROS and the modulation of ROS in the system here. This enzyme, that is P450, can generate ROS of its own, and of course, as earlier I have told you, it can also be generated by the flavin system, the CPR. Right? Now, and further slides we will see the role of CPR as well. And then you have the modulators over here, like cytochrome B5. And now, as soon as the substrate comes in the vicinity and you observe this thermodynamic pull, there is a reaction. There is a quick release of this ROS, and it can just attack the substrate, give you the product. So, this quick and the mild, unrestricted burning that keeps on happening. That's the main understanding of Merban. The burning happens continuously, mildly. That's why Merban, mild, unrestricted burning. It's continuously happening, and as soon as the substrate comes in, it is going to be released and the reaction happens. Now, it's the same thing presented in a little different way, and here we will not call it the classical mechanism. We'll call it the erstwhile, because we have very clearly refuted it. So we'll call it erstwhile hypothesis that you see in your textbooks probably. Now this is dependent on the substrate binding to cytochrome P450, which is not the case when you look at the Merban concept. And then this mechanism requires the binding of not just the substrate to the P450, it also requires the binding of P450 itself to the cytochrome P450 reductase, that is CPR enzyme, like what we have seen here. See, this binding is required in the erstwhile hypothesis, where it's not really required in the Merban concept. So, more facile, better, easier for the substrate hydroxylations or the reactions to actually proceed further. The product from ROS formation requires, and what is the driving force here? Is the thermodynamic pull, not the binding of the substrate, right? So, but more feasible. Thermodynamically, it is a much better explanation than the erstwhile hypothesis. Okay. Now, when you talk about the erstwhile hypothesis, it also mandates that everything happens in the active site. The concept of active site, the lock and key model, or maybe the induced bit model. Either of that talks about the binding of substrate in the active site. So, unless this happens, and everything happens only here, right? So what do you expect when things are happening in the active site? You can get only one kind of a product, and you can only allow certain kinds of substrates. But when you look at the P450 system, you will see a lot of promiscuity. What is that? A lot of substrate, different kinds of substrates. Right? Now, we'll come to that slide in a few seconds. When you look at the Merban model gives you the freedom to understand the binding of substrate that can happen either here this is a substrate, xenobiotic is the substrate. We are talking about xenobiotic metabolism. So it can bind in the active site, or it can bind somewhere near the channel, or maybe some other place from where it can allosterically modulate the enzyme reactions. Right? So this gives you a lot of freedom to explain the kinds of observations in the microsomal metabolism, like the different concentrations, how they affect, and the uncertainty regarding the locus and modality of the ES interactions, etc. So we have been talking about the pull. Now, how is that pull? How did we get one slide? We have shown this here, the pull effect, thermodynamic pull that we talked about, and this slide also talks very clearly, shows that when there is a substrate present, there's this thermodynamic pull. Now this is we are talking about the peroxide in the reaction system, and when you add the substrate, which is the hollow squares. So what do you see? At most of the places, what do you observe? You observe that the peroxide levels are actually down, actually less. Why is that? Because once the substrate comes inside, they are actually being taken up. So there is this thermodynamic pull which is actually driving this whole reaction. So that's why you get this kind of an observation here. Now this has been shown in different reaction systems, like it's just a test tube reaction here. In here you have a bacillosome system, and then you have the CYP-CPR system that we actually talk about, and then the reconstituted systems as well. So almost everywhere we have kind of similar observation. Now, so this is an interesting study where what do we observe here? We talked about the thermodynamic pull, right? Very evidently, when we look at this data, we are actually talking about CYP2C9 reaction, and CYP2C9 reacting, metabolizing diclofenac, giving you 400% diclofenac, which is the product. So this is the standard reaction, and when you add other enzymes, like you have SOD, superoxide dismutase. As we know of superoxide dismutase, it actually works on the superoxides, and it can actually only work on superoxide, and it can take that away. But still, there are other DROS species that will be available. Because there are other DROS species available, you still observe some formation of the product. And when you talk about catalyst can work on peroxide. So still you have some other DROS which it cannot work on, and that's why you still get some level of formation of the product. CP also is a similar case. But when you talk about HRP, CP is a little hydrophilic kind of. It stays in the aqueous phase. So you observe this kind of you still observe reactions over here. But when you talk about HRP, there is a little hydrophobic, and then it takes away all the DROS. It can utilize all the DROS, and because it can utilize all the DROS, it does not allow CYP2C9 to actually hydroxylate any of the any molecule of diclofenac. So we don't observe any product formation over here. It's not driven away. It's not taken to a different direction. So it can only happen when there is a diffusible radical which is going out and doing the reaction. Otherwise, if it is so very active site mediated, this kind of observation is not possible. Because the substrate will go and bind there, and then there will be hydroxylation, and you will eventually get the product. Some or other amount of product you will definitely observe. So we can also see here this ascorbic acid, which is again a soluble form, and then you have the palmitate. The palmitate is again hydrophobic. So when you add the hydrophobic moiety, there is inhibition, quite a lot of inhibition observed, and when microsomal systems are at the membrane, the reaction happens in the lipid phase. It's kind of lipid phase where the reaction actually happens. So a lipid phase modulator, inhibitor, works much better here. Ascorbic acid can also do the job, but then if it is in the soluble state. This is a classic piece of work here from where actually Sir had observed one of the starting points from where the ideas erupted. Now, what do we observe here? There is this reaction system where you have CYP-mediated, the enzyme, the P450, and the substrate inside the membrane, and then it is separated from the P450 reductase. Now, P450 reductase, according to the classical mechanism or the erstwhile mechanism, needs to bind to the P450. Without that, the electron transfer is not going to happen, right? So, what if you separate them? You put a membrane between and you separate them. We are not going to expect any reaction if the erstwhile mechanism is actually correct. But then, interestingly, we not just only observe reactions of almost the same level, but we observe very specific reaction. 4-hydroxyl diclofenac is the product, and we observe that even when you have put the whole reaction in the tube. Why does that happen? The dross that is generated, the DRS that is generated here by the CPR diffuses through the membrane. It can comfortably get there, and the reaction can proceed. This very clearly shows that the reaction is not driven only after binding. I'm not saying binding doesn't happen at all. No, it's okay. It might happen, might not happen. Secondary. But what is driving the reaction? The reaction is driven by the reactive speed, the diffusible reactive speed. So it diffuses through, right? And then it gets inside there and the reaction happens. So now, if it is happening through the diffusible species, next thing was to observe the structures of all these P450s. So we took a lot of P450 system over here, and we tried to observe what is their how they are, in 3 dimensions. How do they look? And there is one common thing that we observe everywhere. We see here that actually, if you look at 2S6 and 2E1, you can also observe a red dot over here and the green dot. So this is actually solvent accessible. There's the proximal side where the cysteine actually holds the heme. This is the proximal side, and that is accessible to the solvent. Invariably, in almost all, except this chloroperoxidase, is accessible in all the systems. Right? And yes, heme is very accessible here in 2E1 and 2S6. We have variations because of that. But of course, just to understand at least the diffusibility, the chances of things getting diffused is not only happening at the distal side, where the erstwhile hypothesis talks about binding of the substrate. That happens, or doesn't happen, it's a secondary stuff again. Now, not only at the distal side, but also the proximal side is actually accessible to the solvent. So it can diffuse. If there is a dross generated, if there is a dross, it can diffuse through this way. And, okay, I'll come to this, continue with the explanation here. Before that is one of the understanding that is required to understand the role of dross in the reaction that it can carry. And then there was another big question. When you talk about, I told you that P450s are very promiscuous. They can metabolize a lot of substrates. And not just a lot of substrates. They can metabolize substrates of different topographies. Now look at this. We have small molecules. We have this big a molecule, we have much bigger a molecule here. All of this can be metabolized by one step. The example taken over here is of CYP2C9 and CYP3A4. This is the diversity of just CYP2C9. We can't just imagine this. If you take the classical understanding of enzyme kinetics itself, what do we know? We know about either the lock and key model that was given by Fisher, or we know the induced fit model that was given by Koshland. Now, lock and key definitely doesn't fit here. Very clearly, it cannot. It cannot explain the binding of such varied topographically varied substrates. Not possible. But when you even take into account the Koshland's induced fit, even that cannot allow the binding of a molecule like trabectedin or retinol in a place where it is going to bind amitriptyline. It can probably give you some space for this, but it cannot just unfold completely and allow trabectedin to get inside the active site. Not possible. But then trabectedin gets metabolized. How can it be? The classical understandings cannot explain it. So, what can actually be happening? This is the plane where this is the heme, and it can take different oxidation states, and then it can have different kinds of the oxygen, like you have oxo, peroxo. All these states are okay, possible. But then what else do we observe here? We observe different channels that are open, from where you can actually access it. And if you look at that, if there are channels here, then, and taking into account Melbourne, if there is a dross generated, it can be released and can be taken up by a substrate because of its thermodynamic pull at the place where it is binding. Okay. So it can be here, it can be in the active site, it can be somewhere here, one opening of the channel, then there can be this one, or it can be the proximal site, it can be either of it. So that's what is shown over here. Like you have trabectedin. Trabectedin cannot bind inside, it cannot. And we have seen this. With docking studies also we have seen, it cannot bind inside, it cannot get inside. So, but it binds on the surface. And where does it bind? It binds very close to the channel. It doesn't bind randomly anywhere. It binds very close to the channel. There, it has a very high probability of undergoing a reaction. Now, this is what Sir was talking about yesterday when he said Moore zone. This is the Moore zone where the reaction can actually happen. The dross comes out, and there is a very favorable chance of finding a ROS that can metabolize substrates. Right? So we have been seeing, very clearly that it's the requirement of the ROS, the diffusible radical oxygen. So, if that's the case, then do you really need the partners that we have been talking about in the way they have been presented all the time? Exclusive partnering, like if you take P450 CAM has its own redox partners. The classical understanding. We change the classical partnering of P450 CAM over here, and then still we can get the reaction very comfortably. We change the substrate. If you change the substrate, you cannot expect the reaction to undergo if it was a binding based mechanism. But if it is a diffusible radical driven reaction, the reaction can still happen if the substrate is present in the Moore zone. That doesn't mean that you can have any XYZ P450 for XYZ any substrate. No, that's not what we are trying to say. You need a Moore zone for that. And a Moore zone depends on several factors. Of course, my talk is limited, we cannot go into that right now. But then, yes, we have seen very clearly that when you are switching partners here, you have 3A4 reacting with coumarin. It's not the substrate actually for 3A4. That also can give you the product. And when you have P450 which is a bacterial P450 system, you allow that thing to metabolize coumarin. Even that can do it. And when you talk about CYP2C9, even that can do it. maybe the efficiencies will change. It will change because there are a lot of other things that are driving. There is a thermodynamic pull, there are other factors also which will affect it a little bit here and there. But then the reactions can happen. So it is not absolute one-on-one binding in that side, only then the reaction can happen. No, it's refuted here very clearly. Yeah, and on top of everything, to just show that it's actually radical species, diffusible species, this reaction, you don't have anything. You just have an enzyme, no redox partners as such. There is no CPR, nothing. There is only superoxide. Superoxide is the dross, one of the dross, right? But it can alone do the reaction. This alone, this system alone can give you the product. So very clearly, this gives you a very strong proof that it is the involvement of ROS. It's not just binding and you need a partner, specific partner. You actually need ROS, not the partners. We had taken this further, and here in this particular slide, what you are observing is the case of benzbromarone, one form of benzerones, which was reported to have, nanomolar levels of Ki values, inhibitory constants, which we could not, bind because when you talk about inhibitory constants, it's actually Ki value. It's actually it's a binding based value. It's not just like IC50. IC50 can talk about reducing the rate of reaction. But when you talk about Ki is actually about binding of the inhibitor to the enzyme. Only then you can get that value. And when you see nanomolar levels, you're actually bringing it lesser than the amount of enzyme that you have provided over there. So even that is lesser than 1-to-1 binding. This cannot explain the kind of inhibitions that benzbromarone was claimed to have. So we took up that thing and then we observed, we did reactions with, and then it was also said that it is a specific inhibitor of CYP2C9. So we took up different systems. We took up CYP2E1 and other enzymes also, and we saw that no, they are not inhibitors of just 2C9, they are inhibitors of other systems as well. Plus, there is a very interesting observation over here. When you look at this, we have, this is the concentration, like you have 100 macromolars of the inhibitor here at this, in this row, and then 1 and then 0.01, that's much lesser over here. So if you talk about a binding based inhibition, this row should have the maximum inhibition. But what do you observe? You also observe, like you look at this particular case, you see here the inhibition is higher than what you observe here. How can you explain that? And there's not just one instance, there are other instances as well. Like here. So, here. So, how can you explain that? If it is binding based? No, sorry, it cannot. Okay. And the non-specificity of the inhibition by benzbromarone that I just talked about is shown here in 2C9 system. It can inhibit 2C9 system. It can inhibit V4 system as well. 2D6 also. 2E1 also. They do not really have the same active site topology. So you cannot imagine, you cannot think of it binding to all these enzymes in the same way. It cannot be an inhibition just by binding. Yes, if it is draws driven, and if benzbromarone can eat up the draws and not allow the enzyme to proceed further for the reaction, then you will observe an inhibition. And that's something Multiple concept helps us to understand. Now here we see the ability of lipophilic molecules to inhibit much better than the soluble ones. That's again because P450 is a lipophilic. The reactions happen in the lipid. So
That's why we see lipophilic molecules to act much better than the soluble ones.
So what do we have here? We have seen that substrate
diversity. We explained the substrate diversity, which the classical mechanism doesn't address too very well.
And then there are so many other effects like secondary oxidations, kinetic
acidosis
effects, then oxygen species binding to CYP, and then also about the electron transfer from the CPR.
We have seen that no, the binding is not protein binding that is always advocated is not really required. So all these things, all of this observation that we have, the different observations which were not actually explained very well by the classical mechanism, Merban has the ability, not has the ability, it actually explains all of them very clearly.
So here we see the role of, we have cytochrome C with the CPR, and then we see when you add the inhibitors like you have cyanide and you have even copper ions, which can, all of cyanide,
we call them as, binders and all, but yesterday itself, we can have talked about it. It must be clear that it's not actually a binding based inhibition.
So,
as the ROS it takes away,
so we see copper ion also
having the ability to do that. So,
and then we have the effect of vitamins and some of the derivatives of the vitamins,
which is like
we talk about this particular graph where you have a soluble form of the vitamin C, and then the lipophilic form of vitamin. So the lipophilic ones are not showing a lot of difference over here in the with respect to the inhibition of cytopropyl, which is a soluble system. So when you put a soluble enzyme, soluble vitamin, you can see the inhibitions,
the variations in the activity.
Now again, what does that actually imply?
It implies it's not really binding, because the system has to be present. I mean, the inhibitor has to be present in the actual zone where the reaction is happening. And
if
the reaction is happening in the active system, it has to be the molecule has to be hydrophilic, or if it is happening in the lipid system, then it has to be lipophilic, right? This is a very interesting observation.
Like when you look at the concentration of an inhibitor,
right, is an inhibitor,
known inhibitor for
the people with the
systems. Now, what do you observe here? We are going down with the concentration here, and then we observe
sudden activation at some position. How can we explain the sudden activation? Again, binding cannot do that.
And then you again see a sharp dip. Higher concentrations, of course,
there are inhibitions, but then there are other instances also where we have seen no inhibition at the higher concentration, and then there is an inhibition at the lower concentration. So if there is no inhibition at higher concentration and inhibition at the lower concentration, that again cannot be explained because of binding. Right?
Like you observe here, like there is an activation rather,
right?
And then it comes down here. It lowers down when you are lowering the concentration. So, and this, okay fine, this is just a reaction, but here the spectroscopic data that we see, the spectrums will actually show you if there is a binding happening or not. If there is a binding happening, there will be some shifts that we observe very clearly. But even with 100-fold excess of 100-fold of the inhibitor present in the system, you don't really observe any shift. So that shows there is no binding. There is no binding as such. Yeah. Now we are looking at a slide which talks about the hormetic effect. And what is the hormesis? Hormesis is basically when you go down
when you lower the concentrations, you observe some activity. Homeopathy talks about that kind of stuff, but the concentrations that homeopathy actually deals with, it becomes doubtful whether there is a molecule in that or not. So, as Sir said, we are not really advocating that part. We haven't gone into that kind of research. But yeah, something like what you see in homeopathy. So, here, when you look at the different concentrations of azide that is given to a growing system, you have Bacillus megaterium, gram positive and a gram negative system, which is E. coli. You do not observe effects on the gram positive, but then when you look at the growth, effect on the growth of the gram negative bacteria, it's very evident here. Like you have micromolar concentrations. This is the when you are having millimolar concentrations, there is a lot of inhibition, and then when you go to micromolar, it kind of normalizes, and then again at nanomolar level it takes a dip. So all this is very weird. But even more weird is when you go a little more down, you go to picomolar levels. What do we observe? You don't observe the 100% growth. We observe even beyond that. That's an activation. And that's the kind of hormetic effect that we're talking about. Picomolars are much lesser concentrations. The controlled reaction rates are shown on the right hand side. You don't observe such kind of thing over here. Yes, of course, at the millimolar level, you have a sharp, very sharp inhibition. Apart from that, no inhibition, no such activation. I mean, there is not, even if we have observed a little bit, I wouldn't call that to be very significant for the thing that we understand. This is again a very beautiful piece of observation where you have two systems. We are talking about one enzyme here, which is the peroxidase enzyme. I told you we are going to deal much less with the peroxidase. We have HRP, oxidative peroxidase, as one of the the model enzyme that we take. And HRP is actually used in a lot of things. Medical uses are also there. A lot of medical uses. A lot of detectors that we use has HRP. The ELISA-based readers. All of them. Even the glucose strip that we use to check whether the blood sugar levels are okay or not, that also uses HRP. So, what do we observe here? We are seeing. Before we get into what we are seeing, now you look at we are talking about the effect of cyanide over here, right? And cyanide, yesterday Vivian has talked about it. Cyanide has a pK value. I don't know if you remember that. It's somewhere around 9.2. So when you get near 9.2, like when you are at 9.2 or higher, you will get better inhibition. So when you look at pH 7, now here an observation, when you look at pH 7, the substrate and the enzyme, take both of them as a couple here, and we have HRP MUTS even here. Now when you look at the IC50 that is observed at pH 7, it's still high, and then when you look at pH 9, it's very, very sharp, very high. And when you look at pH 5 again is coming at 194. So what is the kind of pattern that we are observing? 194, it goes high and then it comes down much lesser. If it is dose dependent, you won't observe this kind of a graph, right? Lesser at pH 5, higher at pH 7. I mean, actually speaking the other way around. Inhibition more at pH 5, lesser at pH 7, and very strong at pH 9. That cannot be explained by a dose dependent. I mean, if it is, even if you take the ionic states, a gradient is not observed here. So the classical understanding of inhibition does not explain this. Further, Vivian has actually talked about it yesterday. So not spend much time over here. One very strong observation. This one, let's talk about this, where you have pyrogalol as the substrate, and then we are changing the concentration of the substrate. Here you have 2 millimolar substrate, so more substrate. To inhibit that, you need more inhibitor. What is the IC50 we are observing? We are observing 10 micromolar. Lesser substrate. You should need lesser inhibitor, but you need actually more. Just the ulta, just the opposite. Here there is no difference. How can that be? You don't have a difference. This is 10. There is much lesser concentration. It's 0.2 millimolar. This is 1 millimolar. But eventually you have the same values of IC50. 193, 194. You take the other one, it's under that concentration. It's almost the same. It's the same value. Cannot be explained again by binding these mechanisms. So we have seen a lot of different observations. Like we just saw hormesis when you have lower dosage, and then at lower concentrations you have activity, higher activity, hormesis. That can be again explained by the Mormon concept. And you have the idiosyncratic responses, where under certain conditions you get weird observations. Like this cannot be just explained by a gradient or some dose response mechanisms. And then all the other like zigzag curves that we have seen, multiple peaks, all of this can be better explained with Mormon concept than it can be explained by any binding based mechanisms. Thank you.
CYPs and dose responses, because as you all know, drug metabolism plays a very critical role in determining efficacy, safety, and therapeutic outcome of medications. And CYPs, a few classes of CYPs especially, they are the major enzymes involved in drug metabolism. So, a lot of examples he explained how these dose responses or the variations, all these can be explained by an effect. So now the session is open for questions from the audience.
Please. Sir, there is a question.
Yeah, you are. Compliment something because I work with the metabolism in live animal. Okay, I also say for field services. As we explain the cognitive responses, everything, but we are unable to explain it. Actually, we are taking that this concept is very easy. Last month I got when CYP-mediated focus, they really need to know about this theory. Okay, then one thing actually is that when you are talking about the RS, it is beginning. Can you use any basic mechanisms for RS and directly find out whether it is really those many places what you are saying is that it will be good because the classical theory doesn't explain it. You are telling that this even in the Mormon theory holds better. Okay, so if the RS can be done, RS can be directly blocked. So, have you done any experiment like this? Because I have not done any in vitro experiments. I have to do in vivo experiments. That is my first question.
Okay. Yes, sir. We have actually shown the results of. Yeah. The slides are not here, sir, right now.
Can you shift to the slides, please?
Yeah. Here also we have done the same thing. Like here, the quenching of the ROS that we are talking about ROS mediated mechanism. So, if you look at this is the basic basal reaction, right? CYP-2C9 is the mechanism that we are dealing with right now over here. And when you are adding a quencher of ROS, right? So SOD is a quencher of ROS. SOD is superoxide dismutase. It will quench the ROS. And it will quench which ROS? It will quench superoxides. Then we have catalase system. Catalase is again a quencher in this particular sense. Of course, this is an enzyme, but it's also a quencher. So it will quench
H2O, peroxides. And then we have HRP. Chloroperoxidase can also do the same job as that of what catalase is doing. But then when it comes to HRP, which also has a hydrophobic nature a little bit, it can quench dross of different kinds. So that's why we observe complete inhibition. So here, because
You see the ascorbate.
I was seeing this like that.
The point is there, you can see.
it's a soluble system, sir, over here, like ascorbic acid. We know ascorbic acid is as a good modulator. It's a very potent redox molecule, but it will work in the aqueous phase better. No, here we are dealing with cyclosinine, which is a lipophilic system. The reaction is happening also. And when you modulate that, you change it. Like we have ascorbic palmitate. Now we have a palmitate chain over there. It can go inside the lipid phase and then it can inhibit it.
Okay.
So that's what we have been trying to project.
When there is just ascorbate, there is no inhibition. So that's site-specific, which place the loss is present, there is stability. So what they thought is when they put SOD, it doesn't inhibit. So they thought loss is not involved. It was an MAH project. While drug is optimized, my job was to see that, my job was to see that we minimize ROS production during the lead drug optimization. So I reported to my professor that if we take out ROS, it doesn't work. So that's what is happening. They put ascorbate, nothing happened. SOD, nothing happened. But the point is to take the ROS scavenger where the ROS is produced. So he showed another system where you see cytochrome C getting the electron from cytochrome P50 reductase, and that electron reduction happens in the aqueous system. There you see the different thing. That's why I said yesterday where the reaction occurs, at what time and in what amount the ROS is. That determines the outcome. The idea that ROS in itself is bad or not involved is not right. This is the reason why we missed out all these observations.
Now, I noticed this disadvantage when we do the in vivo experiments. The animals when we do, we get the same apparent responses. But we were not able to explain it to her because that is a very composite system. At like all the cell groups will be there, all the compounds will be there, everything is composed will be there. When you take the case of animal, because we take raw pure.
We published this in Dose Response. It's an American journal. Edward Galbraith was the editor there. And he was very excited. He said the exact same thing that you said. We have been observing all the while, and we have been sidetracking it as, artifacts. And now we have an explanation. So he was very clear. Exactly like you are.
And one more thing, sir. I think so can you derive equations for like this?
That's wonderful. That's wonderful. We tried a recent paper in Biomedical Research International in 2026, Wiley paper. It's a Q1 journal. We tried to get the mathematical hang of it. As of now, we have got biphasic, zeroth order depletion, all that mathematically figured out. But the complex phases, we will have to go a little more deeper. Wonderful question. Because on that alone rests our ability to predict. See, he showed some, examples of formative things. We started to model that, we couldn't get there. It's a very, very, very hot topic. Because if we can give custom medicine, I have a friend who, when he sits on a plastic chair, he starts coughing. He sits on a steel chair, No problem. And people, they say, this is unreal. When you see our body as an electrostatic system, you can get to there. So there are physical, there are chemical, all this being sidetracked. Sometimes parallel systems of medicine, kind of by hit and trial, get there. So Moodboard allows you a framework where you can mainstream all these things.
Understood.
I'm so happy to see something.
That's a really wonderful thing that we never thought of such a possibility that after coming to UK, you know, this is one thing that is possible, and every theory just to be modeled mathematically, then only it will have the stability. Okay, that is because acceptance will be.
Yes. Yesterday we showed that algorithmically, mathematically I didn't go into because the audience will not follow. That's why.
Okay.
Today we have certain mathematical models we are not going into.
Okay.
Yeah, but your point is absolutely. Michael is mentioned it can be not because it has mathematical ground.
Yes, exactly. You have that thing. Now, another thing is that in various small scales, what happens is the quantum phenomena that actually says quantum phenomena that are setting. How you have thought of such possibility? Because another area I'm working is with the anesthesia.
That's the next topic. Anesthesia will come to.
Okay. So in anesthesia, we have the phenomena we cannot explain by the conventional, the receptor theory. Receptor theory. And the second thing is regarding the mind or consciousness. What about the consciousness?
Mind, I don't know whether we can get there as of now, but then approaching mind is only possible with Moodboard because the classical theory is ionic. This is electronic. So that we'll talk about in the next session. That is neuroelectrophysiology.
Yeah, exactly.
Neuroelectrophysiology. In the next session. It's coming up.
Okay.
We'll discuss. Yeah, wonderful.
Thank you.
That's very good.
That's for me. Okay, wonderful.
Thank you, sir, for very nice question.
Yeah. He's from the field, so.
Very different.
is present in endoplasmic reticulum, but there are non-microsomal enzymes which will be present outside the smooth endoplasmic reticulum. Okay. So they are also making the drugs to metabolize various reactions like many hydrolysis, acetylation. Many are also coming from non-microsomal enzyme part also. So can you correlate the reactive oxygen species generation with the non-microsomal enzyme rather than microsomal enzyme? It can be a futuristic study also if you correlate it with the non-microsomal enzymes. And
We actually have done that. That's why you could see the connection between the peroxidase systems that we had in HRP for that instance. Basically, we took up with HRP and catalase and chloroperoxidase systems, and then from the observations that we got there and the ideas we built upon the understanding in the microsomal system. So it's
actually not microsomal, it is non-microsomal, it is completely different.
non-microsomal. Peroxidases are non-microsomals. All studies peroxidase if you talk about. If you talk about chloroperoxidase, you talk about catalase system. All of them.
There are lots of. He is asking a very valid question.
Can occur in soluble enzymes and all that. So now the point is, the fundamental point that we want to see in xenobiotic metabolism is, it doesn't have an evolutionary memory in your system. Because normal molecules like carbohydrates, lipids, proteins, and all these kinds of molecules, they have evolutionary basis in your system. Their molecular topology is well known. You put a molecule like aspirin, ritonavir, sildenafil, any drug, it doesn't have. So Moodboard offers the first basic scope for how either a polar or a larger molecule or a hydrophobic molecule, all those can be transformed and gotten out of the system. So this Moodboard process doesn't necessarily depend on endoplasmic reticulum stationing alone. It says that diffusible reactive species are generally produced in various locates. If you had come to my talk yesterday, I had shown that. Whether in endoplasmic reticulum, whether in cytoplasm, any locale, whether in peroxisomes, whether in lysosomes, all that, extracellular region, it gets produced. So normal amount of ROS production, there is a sustained release, and then when that sustained release happens, these stochastic interactions with all these at different kinds of molecules at various sites can naturally happen, and that's how they get out of the system. So wonderful question. He was saying that.
One more thing, sir told me large structures, these enzymes, because you showed one track with large structure, you told that the microscopic substrate cannot access inside. But if it starts in a particular,
like if you look at the molecule that we are talking about here, I think you're referring to this particular molecule.
Yes.
Perfect. It's enormous in size. It can never get through the channel also. If you get a deeper look in the at the structures, the structure function relationship, if you look at that, there's no chance that it can actually enter. It's very huge. Even with respect to the channel, even if it opens up a little bit, because you can't expect the protein to completely unfold.
How this drug will metabolize if it is?
That's what we have been talking about. The diffuse, the ROS that is generated, let's say the ROS is generated at the, here, you look at this. Now you have a ROS generated here, at this point, at the heme center. This is a simplified version of the enzyme. This is the whole enzyme. 1 second. This is the enzyme. Now you have a heme center. We know that we are talking about before, so we have a heme center. Now the heme center is somewhere here inside, just that side here, and then at this point you have the dross that is getting. It's also held over there. That's another function of the enzyme that we talk about. But then let's assume the ROS is there. So once it is activated, the ROS is there, or if it gets the ROS from CPR, that we have shown in the other example where we have separated the CPR and the cell system and we have put a membrane. Either of that. You get a ROS over here that is held up, modulated in that particular region, and then once a substrate binds, comes here, like a molecule like trabectin, you can imagine, since it cannot enter, it will be binding somewhere here on the surface. Okay? And the surface is not like blinding some surface. We're not talking about just randomly any place. It should be somewhere close to the channel. Now this channel that opens up, like you see here, the channel opens up here, and very comfortably the ROS that is present inside can diffuse through. It can come and attack the substrate which is bound at the channel. Isn't it? There's somebody huge coming from this, has to come from this gallery, but cannot. What do? You have to send something to that person or whatever. You just let it go through this. It is diffusing through this, and if it finds that diffused molecule that we are sending at the root, and then it attacks, it gets whatever it is. That is what we are talking about.
It can be possible.
It is possible.
This is a very important question you asked. Actually, when I had gone to Gordon Research Conference in 2015 in US, it's a drug metabolism, the biggest conference. Pfizer, Merck, all those people, they conducted. The question, Gabriel Cruciani, he is the supposed specialist to join. He is vacationing, so he is not joining. He just sent me an email yesterday that he has to be away. Gabriel Cruciani is the professor of drug metabolism and all that from University of Perugia, and he has a molecular discovery. He is a director of multiple companies doing this. He was making a talk there. So he was showing how these cytochrome P50s open up and allow these large molecules to go into the active site. He does all those high sophisticated computation. So I just stood up and asked him, don't you think this is all far-fetched? He just stood there and he said, yes. I'm doing it because everybody expressed it. And that's the question that you have asked. Wonderful question. And then came Paul Hollenberg. Paul Hollenberg is a very seasoned scientist from University of Michigan. So he gave a talk where he mutated surface residues at different places. Surface amino acid residues. So he saw that the CYPs failed to metabolize. So how do these surface residues mutations affect catalysis? So this Moore-Burn explanation gives a very simple explanation. You see, that's what Abhinav has drawn there. There is a catalytic active site in the center, just above the heel. The molecule, it's like an elephant cannot come through this. But like he explained, the small rods can definitely go there. The molecule can bind there, there. If this is the protein, right? That's what he explained. Wonderful. Nice explanation.
Ideas interaction. Just speak out aloud some of the thoughts which I got on the basis of listening to Parashar. I'll focus on diabetes mellitus and the butyrocholinesterase enzyme 1. You've done a lot of work on the mitochondrial pathological changes, the reactive oxygen species. I had been to one of the GARC conferences, a 3-day conference on mitochondria and their role in diseases. Maybe if you could apply the Metabolism concept to the oxidative stress or the ROS and diabetes mellitus insulin resistance, that's a potential area in which you can work. That's one thing, maybe some of the junior colleagues or the scholars can work on that. And second is, if you could just see whether butyrocholinesterase is a neurozyme. Yesterday there was a deep learning analysis of this. There are 40 variants of this enzyme. And if you are interested, you could just look whether any of these fit into the neurozyme. And you also showed that this drug metabolizing enzymes can fit into many pockets. So see whether butyrocholinesterase or its variants can do that. And finally, we have data on raw data on butyrocholinesterase sequences along with its variants. If some of you would like to do molecular dynamic studies, I'll be able to share it with my computer sciences colleague as well as you. These are some of the thoughts which I got as I was listening to you.
Thank you, Dr. Sridhar. Thank you, sir. I think we are. Thank you, sir.
So, thank you all. Thank you, Speaker Abdul, and then all the wonderful interactions. Thank you, Dr. Sridhar.